Hello, my name is Rick Weinzierl. I'm a professor and extension specialist in the Department of Crop Sciences at the University of Illinois. This presentation in our Beginning Farmers program for fruit and vegetable producers focuses on plant disease management. Darren Eastburn and Suzanne Bissonnette of the University of Illinois contributed slides for this presentation. As a result of watching and listening to this presentation, you should be able to develop an increased awareness of different types of diseases and disease-causing organisms. You should understand the key practices that prevent or reduce the likelihood and severity of plant disease outbreaks. You should know the major diseases that warrant concern in vegetable and fruit production in Illinois and where to find recommendations for their management. Your longer-term goals are to develop plans for disease management in your own crops. That step will require lots more time and effort and will require constant updates and revisions and attention. Remember that disease management is a component of overall crop management and integrated pest management, or IPM. In an earlier presentation, I pointed out that IPM is an approach that uses a range of practices that limit losses to pests while minimizing the environmental damage, human health risks, and dollar costs associated with pest suppression. Tactics include biological control, cultural controls, resistant varieties, regulatory programs, and pesticides where needed and in ways that minimize their adverse effects. Remember that in IPM, pests are broadly defined to include weeds, insects, mites, plant pathogens, and even wildlife. So what is a plant disease? It's a condition in which a plant differs from a normal or healthy state, and it occurs over time. The diseases shown in this slide are tomato spotted wilt virus, black rot of grape, and Phytophthora blight and fruit rot of pumpkin. In our discussion of integrated pest management, I used Basic Concepts in Plant Pathology by John Hartman as a good general introduction to the management of plant diseases. It discusses non-infectious versus infectious diseases, types of pathogens, symptoms and signs of plant diseases, and various approaches to controlling plant diseases. Let's use this approach and add more details in several categories. There are two types of plant disease in general. Non-infectious disease is caused by non-living factors. A non-infectious disease cannot be transmitted. For example, drought, flooding, chemical injury, nutrient stress, and environmental stress are all non-infectious diseases. Infectious diseases are caused by biotic living organisms. They can be transmitted. So air pollution injury is an example of a non-infectious disease, and a stem rot caused by a fungus is clearly an infectious disease. Some examples of non-infectious diseases that are common in fruits and vegetables: glyphosate injury to apple trees. Roundup is glyphosate. Sun scald on pepper, and hail damage on apples. The kinds of plant pathogens that cause plant disease include fungi and fungus-like organisms, bacteria, viruses, nematodes, and phytoplasma-like organisms. Phytoplasmas are bacteria-like organisms that have a cell membrane instead of cell walls and can assume a variety of shapes. They are parasitic only in plants. In many plants, phytoplasmas invade cells, and the food-carrying tissue known as phloem in the vascular system of plants, and they're usually spread by plant-sucking insects such as leafhoppers. They draw their food from the phloem of plants. Phytoplasmas cause some 200 plant diseases affecting several hundred genera of plants. And that's about all we'll say about phytoplasmas and phytoplasma-like organisms in this discussion. So fungi. think you have a basic idea of what a fungus is, and of course tiny microscopic structures make up those fungi. Hyphae that are the growing tissue, spores that are reproductive, and sometimes some resting structures that allow the fungus to persist in the soil for multiple seasons. The ones that infect foliage are spread by rain and wind, and examples of fungal diseases include rusts, cankers, leaf spots, smuts, and root rots. Apple scab is pictured in the slide. It's caused by the fungus Venturia inequalis. A fungus disease only recently discovered in North America is downy mildew of basil. It's a host-specific pathogen, caused by a host-specific pathogen, a fungus that infects only basil. It grows best under moderate to warm temperatures, but not extremely hot. It causes yellowing of leaves, fuzzy white to purple underside growth of the fungus, curling and browning and blackening, and lesions on the leaves. It can be confused with nutrient deficiency early on. It is managed by planting disease-free seed. Some varieties are somewhat resistant. You minimize leaf wetness by watering practices. Scout regularly, and you can use fungicides to control it. The spores in the photo at the lower left allow confirmed identifications of this fungus in places like the plant clinic or a plant pathologist's lab. The picture in the lower right shows what little is left of basil stems and leaves a few days after initial infection. Bacteria are single-celled organisms that typically infect plants by entering wounds or openings in the plant leaf or stem surface. They cause wilts, blights, and soft rots of fruits or roots. Pictured in the slide is fire blight of apple, caused by Erwinia amylavera, a bacterium. Three bacterial pathogens cause spots on tomato fruits, and they are very similar. Bacterial canker, caused by Clavibacter, bacterial speck, caused by Pseudomonas syringae, and bacterial spot, caused by Xanthomonas campestris. These are all similar spotting diseases on fruits, but they cause other symptoms that are not the same from species to species. There are no treatments for bacterial canker once it's established in a planting of tomatoes, but management for speck and spot are quite similar. In all cases, recommendations call for planting disease-free seed, planting less susceptible varieties. There are no completely resistant ones. Minimize canopy and foliage moisture by plant spacing and by irrigation practices. You do not handle wet plants because that spreads the bacteria. You rotate crops to avoid fields where the bacteria is built up or is left over in plant debris in the soil, and by regular scouting and protectant commercial bactericides that can be applied to plants. Again, these are very similar diseases, but they do differ in significant ways. In canker, older leaves become scorched. It can be localized to just one side of a plant, and it causes sudden wilting. This is the only disease that causes, of these three, that causes vascular discoloration in cankers, and the fruit itself shows white spots with raised dark lesions, sort of like a bird's eye appearance, and they're about 3 millimeters or 1/8 of an inch in diameter. This disease is spread by splashing, but also by contaminated seed. Bacterial speck and spot are quite similar. In bacterial speck, the lesions are less than 1/16 of an inch in diameter. They do have small halos, or white areas around them. They may be raised, flat, or sunken lesions on the fruit. They're very small, 1 millimeter, or about a 20th or a 25th of an inch in diameter. This one occurs most often in cooler temperatures and humidities and high humidities, and it's spread by splash from the soil surface. Bacterial spot causes slightly larger, irregular spots on fruits and leaves, sometimes with halos that and some scorching or some overall browning of foliage, as you see in the lower right. Fruit symptoms start as tiny spots, about 1/8 of an inch long, but they become larger with raised scab-like brown lesions around them. It's favored by warmer temps and by precipitation, and this disease also affects pepper. So these are just examples of bacterial diseases of a very commonly grown vegetable crop, tomatoes. Plant pathogenic viruses are typically small fragments of RNA. They cause spotting and mosaic or mottled patterns on leaves or fruits, and they reduce plant yields and produce quality. Some are seed-borne. Viruses may also enter plants as a result of physical damage to leaves or stems, and insects, nematodes, and mites may move them into plants as they feed. By far the most common, but not the only vectors of plant pathogens among insects, are aphids. And aphids transmit cucumber mosaic virus, the symptoms of which are seen in this slide. Nematodes are microscopic roundworms. They may infest or infect plants depending on how you view those two terms. Although some live in foliage or stems of plants, such as pine wilt nematode that lives in the vascular system of certain pines and causes pine wilt disease, most infest roots of plants. Soybean cyst nematode is a common one that attacks soybeans, and root knot nematode attacks carrots and other root crops, as seen in this slide. In the IPM presentation, I introduced the idea of monocyclic versus polycyclic diseases. One reason to understand disease cycles for plant pathogens is to identify the stages in the disease cycle that allow steps to manage the disease and reduce crop losses. Peach leaf curl is one example of a disease characterized by a monocyclic or simple interest disease cycle. Fungi in leaves that show the distortions associated with this infection produce inoculum that reaches the buds that will produce next year's initial leaves and fruits. When those buds swell in the spring next year, the fungus infects the new tissue. Only the fruits and the first flush of leaves are infected in the spring. Later leaves are not infected. Where fungicides are used to prevent leaf curl in peaches, they are applied in the fall after leaf drop or in the early spring before bud swell. Gray mold of strawberries and several other crops caused by a fungus in the genus Botrytis has a polycyclic or compound interest disease cycle. It causes the most damage when rainfall or irrigation during bloom and fruit development allow the pathogen to grow. It infects senescing blossoms first, and infections spread from blossoms to fruit. Spores are produced on infected tissue and on plant debris, and direct infection of mature fruit can occur a few days later. Knowing that senescing blossoms are an initial infection site can guide decisions on the use of fungicides at bloom during periods of rainy weather, and understanding that infected tissues produce spores that can start new infections guides additional decisions about disease management, be it by fungicide use or other methods. We often stress monitoring or scouting to make decisions on insect and plant disease management, and these steps are very important. Even so, most disease management strategies are preventive. Either we use cultural practices that limit the likelihood of disease development, or we apply fungicides or bactericides before infections start. These preventive steps are essential, especially for organic growers. And if we apply fungicides or bactericides to prevent, not cure infections, then why do we scout? Well, for polycyclic diseases, early observations of disease then guide the steps that we take to prevent further infections. Removing infected plants or applying a plant protectant chemical are the steps we then follow with. Just as important, however, is the fact that scouting provides a guide to future cropping decisions, either a longer rotation, the use of a variety resistant to the specific disease, plowing under crop residues, and so on. so observing and recording the incidence and severity of specific diseases are important steps in disease management. So let's return to Dr. Hartman's list of plant disease control practices: avoidance, exclusion, eradication, resistance, protection, and therapy. Several farming practices fit into more than one of these categories, but all are meant to somehow slow the rates of infection and plant disease caused by pathogens. They reduce the area under the disease progress curve, as it's described by plant pathologists, in ways similar to that shown in the graphs in this slide. They illustrate the value of using resistance genes in the management of potato late blight. In most cases, disease management practices do not have to eliminate or prevent all disease. They need to slow its progress so that profitable yields of high quality can be harvested. To avoid pathogens, crops may be grown in regions where certain pathogens do not occur or are rare. For example, several vegetable seed crops are grown in the arid West, where key bacterial diseases do not occur, and that practice produces disease-free seeds. Crops susceptible to aphid-borne viruses are grown early in the season when possible because aphid populations are usually lighter then, and crops susceptible to root rots are not grown on low, wet, poorly drained soils. Crop rotations are designed to place susceptible crops in fields where specific pathogen levels have declined in the absence of crops in that plant family. While crop rotation is commonly understood to provide benefits for vegetable production, it is also important for small fruit crops such as strawberries and brambles, and for avoiding what are called replant diseases in apples and peaches. Seed and plant certification programs, quarantines on the movement of infected plant materials, and the use of row covers that exclude insect vectors of viruses are all examples of exclusion of plant pathogens from crops. Site selection is also an important avoidance practice. Crop rotations and considerations of disease history guide selection of planting sites for specific crops, and so do environmental conditions. Avoiding low areas with poor water drainage is a common recommendation. Similarly, for perennial fruit crops, avoiding areas with poor air drainage helps to reduce losses to freezes and frosts, but also to fungal diseases that thrive in high moisture conditions. We often also recommend that disease avoidance includes modifying the environment. Steps that modify the environment include water management, so that irrigation avoids prolonged periods of leaf wetness. That may mean overhead irrigation at the right times of the day, or trickle irrigation that does not put water on leaves at all. We also use consistent watering so that there are no periods of extreme dry and extreme wet conditions to manage blossom end rot in tomatoes and peppers. We may orient rows so that airflow down the rows helps with drying leaf tissue, but plant densities and pruning are especially important for this. Air movement reduces leaf wetness, so pruning trees so that they are not so dense, or even pruning crops such as tomatoes so that foliage is not so dense, helps to reduce leaf wetness and plant disease. We use mulches to reduce splash dispersal and protect fruit from soil-borne pathogens, and those mulches may be black plastic or they may be plant material, such as miscanthus or straw. And growing in high tunnels is somewhat the ultimate in avoiding disease by modifying the environment, because there is no overhead water applied to plants, and temperature fluctuations are muted. Eradication of pathogens, or more accurately the reduction of inoculum to low levels, is the goal of destroying or burying or effectively composting crop residues, and of crop rotations so that soil pathogens die off or are attacked by other soil microorganisms during the years when susceptible crops are not grown in those fields. Fungicides and bactericides may be applied to plants to protect them from infection. Steaming and soil fumigation, shown in this slide, are not used commonly in Midwestern production systems, but crop rotation is a valuable standard practice for reducing inoculum. Crop rotation again is the most commonly used practice for reducing inoculum in vegetable production. It's most effective against pathogens that survive in the soil or on plant debris, so that during the period between crops that are susceptible to that pathogen, survival declines year after year. It's most effective against pathogens that do not survive well as saprophytes. That means the organisms that grow on dead plant debris. It works best against pathogens with a narrow host range. For example, rotations to avoid problems with sclerotinia or white rot are sometimes tough to accomplish because that disease infects cucurbits, tomatoes, green beans, sunflowers, and a few other crops. And again, crop rotation works best on pathogens that do not spread over long distances by wind. We generally say rotate among plant families so that the same crop is not planted on the same ground for at least 3 to 4 years after the current season. Effectiveness of rotations is reduced if equipment or runoff or wind-blown rain carries soil and pathogens from one plot or field to another. An excellent reference on crop rotation, and in fact specific rotations for specific disease management goals, is Crop Rotation for Organic Farms. And this publication is available for purchase, but it's also available for free download from the SARE Learning Center, and the link for that site is on this slide. So, examples. Among many plant diseases that are managed effectively by crop rotations, root knot nematodes in carrots and related crops, and black rot, a bacterial disease of crops in the cabbage family, are very susceptible to crop rotation. The pathogens decline during periods when the susceptible crops are absent. Crop rotation is not effective for management of powdery mildew of cucurbits because spores of the fungus survive long-range dispersal by wind. Likewise, rotations do not reduce the incidence of cucumber mosaic virus in peppers and many other plants because aphids carry the pathogen from plant to plant over considerable distances. Other practices that reduce pathogen survival include the use of cover crops and soil amendments. Roots and residues of some cover crops promote the growth of soil bacteria and fungi that inhibit plant pathogens. The resulting soils are called disease-suppressive soils. Plants in the mustard family, when flailed and incorporated into the soil, act as biofumigants when they release glucosinolates that kill certain pathogens. Composts, often applied to the soil surface or incorporated, may suppress plant pathogens or provide a physical barrier to rain splash of soil-borne pathogens. Cover crops and amendments that do not directly influence pathogens may improve soil tilth, water-holding capacities of soils, and nutrient-holding capacities of soils. All of these benefits may help plants produce crops even in the presence of some infections or other stresses. Using disease-resistant varieties is often a key step in disease management for fruits and vegetables. Resistance is often the easiest management strategy. It's cost-effective, generally consistent, it's easily integrated with other management options, but resistant varieties are not available for all crops and all diseases of those crops. In general, however, we always recommend to use resistant varieties as much as is practical for all vegetables and also for apples. You do have to have a market for the specific varieties that you produce, but if that is the case, if you can market the resistant varieties, they are especially important for organic growers who will then have fewer options to slow disease development with fungicides or with bactericides than what conventional growers might have. Cornell's Vegetable MD site provides extensive listings of cultivars resistant to specific diseases. And seed company catalogs also list the disease resistance characteristics of their seed lines. The link next to the picture of Crimson Crisp apples provides a listing of disease-resistant apple cultivars. This is just the beginning of the listing of disease resistance characteristics of tomato cultivars provided on the Cornell Vegetable MD site. This portion of the list makes it only partway through the varieties whose names begin with A, and it continues all the way through the alphabet. There are similar listings for other vegetable crops that show their disease resistance characteristics. All are on the Cornell Vegetable MD site. Fungicides and bactericides may be applied to plants to protect them from infection. All products applied to commercial food crops must be EPA registered or exempt from classification because they have been categorized as generally regarded as safe. For organic production, disease control chemicals must be approved by the National Organic Program and carry an OMRI-approved label. The safety of fungicides and bactericides is evaluated in part in comparison with the risks that mycotoxins and bacterial toxins pose to food safety. Diseased produce rarely is linked to foodborne illness, but produce with bad spots from decay does contain microbial metabolites, microbial compounds produced by the pathogen, in portions of the fruit or the vegetable that may appear undamaged, and several fungal metabolites are known to be toxic to humans. So what are fungicides? They're chemicals used to kill or prevent the growth of fungi, specifically plant pathogenic fungi. They are described by their mobility in the plant. Are they contact or systemic? Systemic ones move through the vascular system. Their role in protection. Are they preventive or curative? Most are preventive. Their breadth of activity. Does it work against a single metabolic site in the fungus or multiple sites? Newer compounds with single site activity are more likely to trigger resistance in plant pathogens. They're categorized by their mode of action. If you remember in our discussion of pesticides, fungicides are labeled with a FRAC code, a Fungicide Resistance Action Committee code that guides efforts to avoid fungal or bacterial pathogens developing resistance to the chemicals used to control them. They're also categorized by the type of chemical. Are they inorganic or organic compounds that contain carbon? There is a link on this slide to a site that provides a discussion on the American Phytopathological Society's page about fungicides in general. In addition to the fungicides widely used by conventional growers that are synthetic, copper fungicides are used for disease control by organic growers, and the link here to a Penn State University site describes several of these copper fungicides and their use. Bactericides include antibiotics, biological compounds that induce plant resistance, and copper compounds. The antibiotics, streptomycin and oxytetracycline, are used for fire blight control in apples. The living organisms or compounds produced by them that induce plant resistance induce what is called systemic acquired resistance. And there are copper compounds, similar to the ones we mentioned for fungicides, that are used for bacterial disease control. A good general reference on conventional and organic fungicides, bactericides, and nematicides is available at this link from the Pacific Northwest Plant Disease Control Handbook. It lists trade names, chemical names, the type of activity. Does it control bacteria, fungi, something else? The FRAC code for resistance management, and a general description of uses. Again, this reference is quite valuable for understanding who are some of the fungicides out there and some of the bactericides, and how are they used. In recent years, several new plant disease control products have come to market for use in organic production. They vary tremendously in effectiveness when evaluated in controlled trials. In this evaluation of effectiveness for control of diseases of tomatoes, disease severity did not differ from the untreated control in plots treated with the products listed in the white portion in the top half of this table. So those were not really very effective at all. Bordeaux mix, copper sulfate, lime, and water, was among the most effective of the OMRI-approved products, but several were completely ineffective, as I mentioned a moment ago. Disease incidence or severity in all of the tomato plots treated with these fungicides did not differ from the untreated check. Individual OMRI-approved products may be much more or less effective against different diseases, just as synthetic fungicides may be. Several of the treatments in this evaluation were most effective against powdery mildew and squash. Those included Serenade plus copper, a sulfur fungicide, and even whole milk, which has been shown to work against powdery mildew. Be sure to check the effectiveness of specific products against specific diseases before using them. As I mentioned with pesticides in general, labels may often exaggerate what a compound is actually effective at controlling. Research in the previous table included evaluation of compost teas for powdery mildew control. Although applying properly aged compost to soil provides several benefits, including plant disease suppression, the effectiveness of compost teas for plant disease control remains poorly documented. A compost tea is brewed by suspending compost in a mesh bag in water. Water may be aerated with an aquarium pump or non-aerated, and some recipes call for using additives such as molasses to fuel the development of the tea. There's significant batch-to-batch variation in compost teas, even made from the same compost pile, and there are inconsistent results for these products for disease control. Probably the biggest concern for compost teas is that they may contain human pathogens or at least compounds that are toxic to humans that have been produced by fungi in the compost. The National Organic Standards Board Compost Tea Task Force recommendations from about 10 years ago state that you should use only potable water to make compost tea or dilute it. You should sanitize all the equipment used to prepare compost tea, and make compost tea only from compost that has maintained a temperature of at least 131 degrees Fahrenheit for 3 days and has been mixed so all of the pile or windrow has heated up. You are not to use additives when fermenting compost tea, as these can promote the growth of harmful organisms. In particular, simple sugar sources like molasses should be avoided. If compost tea is made with additives and it's not tested for pathogens or toxins, it should not be applied to food crops within 90 to 120 days of the time they will be harvested. This is similar to the restrictions against the use of raw manure. For effective disease control with fungicides or bactericides, thorough spray coverage is essential. A few fungicides are systemic, that is, they move in the plant's vascular system, but most are not, or they do not provide effective disease control unless plant tissues are uniformly covered. To achieve adequate control of plant surfaces in crops with dense canopies, use an adequate amount of water. often at least 30 gallons per acre for vegetable crops, and at least 100 gallons per acre for mature fruit trees. Spreaders and stickers, that are adjuvants added to the spray tank, may be recommended on the pesticide label to reduce the surface tension of the spray mix so that droplets spread across plant surfaces, or so that the pesticide adheres better to plant surfaces after the water evaporates. For crops with dense canopies, air blast or air-assisted sprayers are used to move the spray into the plant canopy. Regardless of the crop or type of sprayer, using water-sensitive cards to measure spray coverage is recommended. Place the cards throughout the plant canopy when foliage is dry, make the pesticide application, then check the cards to see how well they were sprayed. Water droplets cause the cards to turn blue. If the cards were not sprayed uniformly, then neither were plant surfaces. In general, spray timing must precede infection. For compound interest diseases such as gray mold, sprays may be applied during bloom to prevent blossom infection, and sprays may be needed again after some blossom infection occurs to prevent spores produced on those blossoms from infecting later fruit. So repeated sprays to prevent later spores from infecting leaves or fruit is common for polycyclic diseases such as powdery mildews, downy mildews, several others. On the topic of spray timing, we know that most plant disease progresses more rapidly in warm, wet weather. Predictive models based on temperature, leaf wetness, and other factors are available to guide monitoring and control programs for fungal and bacterial diseases. Some are available at no charge online, others require purchase of weather monitoring equipment and computer software. As I said in an earlier discussion, even if you're not ready to invest in these programs, it's never too soon to read about them and learn about how they can help refine decisions on disease control. Predictive models that help with spray decisions and timing are available for scab, fire blight, sooty blotch and fly speck of apples, for alternaria leaf blight, gummy stem blight, and anthracnose of muskmelon and watermelon, and for the other diseases listed in this slide, or the other crops listed in this slide. Therapy, the removal of infected plant parts or killing off an infection within a plant, is usually not an option. Pruning out fire blight infected shoots, well below the lowest visible evidence of infection, might be considered to be therapy. But perhaps a more widely used version of therapy is hot water seed treatment. Use the link presented here for more information. Several people in our classes have asked about hot water seed treatment. These are temperatures and durations of hot water treatment for several crops. There are a few important points here. Treating seed from commercial suppliers is usually not necessary. And although some references recommend using hot plates with magnetic stirrers along with calibrated thermometers to maintain specific temperatures, others recommend relying on a precise water bath to reach and hold the necessary temperature. Because if it's too hot or held too long, the treatment can kill seeds. If it's too cool or not held long enough, some pathogen survival may occur. And as you move to the hot water baths, this becomes a little more expensive than many of you might choose to do for small scale or small amounts of seed to treat. So what diseases should you be concerned about? Well, there are many of vegetable crops and fruit crops in Illinois. But not every disease listed in every disease control guide around the country actually occurs here. Pages 67 to 68 of the 2014 Midwest Vegetable Production Guide list the major diseases of Illinois vegetable crops, along with information about the roles of tillage, seed transmission, crop rotation, and resistant varieties in disease occurrence and management. For fruits, the most common diseases encountered in Illinois production are listed in this slide. For apples and pears, apple scab, cedar apple rust, powdery mildew, fire blight, sooty blotch, fly speck, and summer fruit rots are among the most common. For strawberries, Botrytis blossom blight and fruit rot, leather rot, anthracnose, and black root rot are concerns. And you can read the list of common diseases for other fruit crops. The same references I have mentioned in presentations on pesticides and IPM provide summaries of what works against what problems. The Midwest Tree Fruit Spray Guide and the Cornell Production Guide for Organic Apples are references you should have for tree fruit disease control. And we'll walk through how to use the spray guide during our class sessions on this topic. For small fruits, use the Midwest Small Fruit and Grape Spray Guide or the Organic Production Guides from Cornell. And for vegetables, the Midwest Vegetable Production Guide and Cornell's Organic Production Guides are very useful. Be sure to look through the general information on vegetable disease management on pages 66 through 72 of the 2014 Midwest Vegetable Production Guide. For additional background, Cornell's Vegetable MD site provides many useful fact sheets, illustrations, and management recommendations for a wide variety of vegetable crops. Similarly, the Midwest Small Fruit Pest Management Handbook and the Midwest Tree Fruit Pest Management Handbook are available online and in print. They provide background information on disease cycles and cultural control practices for many common diseases. In many instances, you will want to use Google or a similar search engine to look for disease identification and disease management guides for specific crops. The two tomato disease publications listed here are examples of the kind of information available online. In many instances, the online references will be most valuable if there are those from .edu or .gov domains, publications produced by university research and extension personnel and by USDA researchers. Good references can be found from private sources, the .com and .org domains, but some of these also advocate for certain commercial products or contain some inaccurate or unsubstantiated claims. So just be a little careful as an information consumer. The University of Florida's site on disease diagnosis is very helpful as well, and you can use the link provided in the middle of this slide. And again, Cornell Vegetable MD, very, very good reference. And a point that we stressed in our general discussion of IPM. Although online and printed disease identification guides can be very helpful, it's often necessary to send samples of diseased plants to a lab where a definitive diagnosis can be done. The University of Illinois Plant Clinic provides such a service for a small fee. The Plant Clinic website provides information on its services and fees and how to prepare and send samples. Planning and carrying out disease management practices that can resolve a problem or avoid it in the future depend on correct diagnosis. Practices that reduce losses to one disease may not be at all effective for another, whose symptoms may look quite similar. So paying a small fee for an accurate identification and diagnosis is almost always a very, very good investment. Some additional information sources include the Resource Guide for Organic Insect and Disease Management from Cornell, the Organic Management of Vegetable Diseases series, Parts 1 on soil and Part 2 on foliar diseases from Florida, and Plant Disease Management for Organic Crops from the University of California. My common recommendation for all aspects of IPM, Read the relevant newsletters that cover fruit and vegetable production. Newsletters from Illinois, Purdue, and Ohio are especially useful for Illinois growers. And the University of Illinois Extension's Local Food Systems and Small Farms team has organized and delivered numerous webinars that are especially useful for new growers, and these discussions are archived and available. And you can see the kinds of topics covered by the webinars that are available from the Small Farms team. Many presentations on fruit and vegetable disease management are available as PowerPoint PDFs on the Illinois Specialty Growers Association website. Program presentations from 2012 through 2014 are available via links on this site. Finally, a few take-home messages. The first would be, think like a plant disease. If you understand disease development, understanding disease control is much easier. So use available references to understand disease cycles, ways to intervene in those disease cycles, ways to meet the challenges that plant diseases pose. Always plan ahead. Use preventive cultural practices such as crop rotations, site selection, planting date, resistant varieties. These are always important. Keep up to date through newsletters and other references. Use the background information to learn how to scout and learn how to manage diseases. Get positive identifications when you don't know what a disease is. And respond effectively, be it by fungicides or bactericides or by altered cultural practices. Always document disease occurrence and history because those records provide the information you need to plan for the future. Always be a little skeptical of untested rescue treatments. Labels for pesticides are sometimes a bit more ambitious than the products actually turn out to deliver. And always consider disease management to be part of an integrated system where you use multiple management strategies to produce a crop with minimum losses to diseases, to insects, so that integrated management of the crop is the framework in which disease management is practiced. If you have questions about the information in this presentation, please contact me, Rick Weinzierl. If necessary, I'll connect you with another specialist to answer specific disease management questions. And Suzanne Bissonnette is the director of the University of Illinois Plant Clinic, and she can answer your questions about using the clinic's services.